In this section, we will discuss about how the glomerular filtration takes place through the glomerular capillaries. This is the renal corpuscle. Now the blood that is about to be filtered, as we have already discussed, it enters the efferent arteriole. This efferent arteriole is actually from the renal artery. Now the blood flows towards the glomerulus for filtration. That is the main site of filtration. It takes the blood from the efferent arteriole and filters it. Now the rest of the blood flows on into the efferent arteriole. Now this filtrate then leaks out of the glomerulus into the Bowman's capsule, into its open space. That is, the Bowman's space collects this filtrate and passes into the proximal tubule, where it is further processed to form the urine. Now the process by which this glomerular filtration occurs, it is called renal ultrafiltration or glomerular ultrafiltration. Now this ultrafiltration means that it is simply filtration that occurs under pressure. Now due to that pressure, the blood plasma undergoes filtration. Most of the particles will get filtered because it works at the molecular level. And also the blood plasma has to move through a filtration barrier to get filtered. So that is the glomerular ultrafiltration. Now about 180 liters of fluid, they are filtered through the glomerular capillaries into the Bowman's capsule every day. But most of this filtrate is reabsorbed and leaving behind only 1 liter of fluid that is to be excreted each day in the form of urine. Although this renal fluid excretion rate is highly variable because it depends upon the fluid intake. But why is there fluid leaking into the glomerulus and filtration happening here? And what's so special about this part of the corpuscle? To answer this question, we have to look at a detailed structure of the glomerular capillary, because the physical characteristics of this glomerular capillary wall, it actually determines that what is filtered and how much it is filtered into the glomerular capsule. So the type and amount of the filtrate actually depends upon this physical characters of the glomerular capillary. So let's have a detailed look at the structure of the glomerular capillary. The glomerular capillary membrane is similar to that of the other capillaries, except that it has 3 layers, and these 3 layers together they make up the filtration barrier. And this filtration barrier, it is the main site of filtration, and it acts as a highly specialized filtration interface, which means that it actually displays high conductance towards the small and mid-sized solutes in the plasma, which means that small sized and mid-sized solute, they can easily pass through this filtration barrier, but it retains its relative impermeability to the macromolecules such as blood cells, etc. So working from the inside out, this capillary wall consists of the barrier, it consists of 3 layers. First layer is the endothelium, or it is also called fenestrated endothelium. In Latin, the word fenestre means windows. So actually this endothelium, it is fenestrated endothelium, which means that these endothelial cells, they are perforated by thousands of small holes or perforations, and these small holes or perforations are called as fenestrae. So that is why this endothelium is called fenestrated endothelium or perforated endothelium. And these fenestrations are around 70 to 100 nanometer in diameter. So that is freely permeable to everything like water, solutes, and small amounts of plasma proteins can easily pass through this endothelium, but not the blood cells or the molecules that are greater than 100 nanometer. They cannot pass through this fenestrated endothelium. And due to this fenestrations, that is the reason that these capillary membrane, these are 50 times more permeable than the other tissues. Also, these endothelial cell proteins, they have fixed negative charges, and these negative charges, they inhibit the passage of large plasma proteins or the negatively charged compounds. The second layer is the basement membrane. This basement membrane is a membrane that is fused to the endothelial layer, which means that it surrounds the endothelium, and it contains the negatively charged glycoprotein. That is why it is considered as the main site of ultrafiltration, because it inhibits the filtration of plasma proteins out of the bloodstream. Now the last layer of the filtration barrier is the podocyte. it is the final layer of the filtration barrier, and it is a layer of modified epithelial cells which are called podocytes. These podocytes, they have long foot processes that are called pedicels, and these cells are attached directly to the basement membrane by these foot processes or these pedicels, which means that they wrap around the capillaries or encircle the outer surface of the capillaries, but they also leave small slits in between them, or you can say that they are separated by regular gaps which are known as filtration slits or slit pores, and through these filtration slits the glomerular filtrate actually moves. These podocytes also have negative charges, and these negative charges, they provide further additional restriction to the filtration of plasma proteins. So all these three layers of the filtration barrier, they provide a barrier to the filtration of plasma proteins and blood cells, but they will permit the rapid filtration of water and most of the solutes in the plasma. Let's have a look at the diagram of the glomerular capillary membrane. This is the filtration barrier, which consists of endothelial cells, which are fenestrated, having small fenestrations through which the filtration takes place. This is the basement membrane that is fused to the endothelial cells. These are the podocytes, which are modified epithelial cells. And these podocytes have these foot-like processes which are called pedicels, and these pedicels also have filtration slits in between them. So these podocytes actually wrap around the basement membrane through its foot processes or pedicels. Now, as you can see in this diagram, it appears as an octopus that wraps around the capillary membrane through its foot processes around the length of the membrane. The permeability of this filtration barrier, it is dependent on the molecular size of the substances. So the substances having a molecular weight of less than 7,000 daltons, they can pass freely, such as ions, urea, glucose, amino acids, etc. But larger molecules are highly restricted up to 70,000 daltons, and above which the filtration is insignificant. So small molecules are freely filtered and pass the filter without any hindrance. The permeability is also dependent on the electrical charges on the molecules, because they also play an important role in affecting the rate of filtration. And the negatively charged molecules, they are further restricted because they will be repelled by the negative charges in the filtration barrier. So the albumin, which has a molecular weight of around 69,000 daltons and is also negatively charged, it is filtered only in minute quantities. So that means that the glomerular filtrate is almost protein-free and is free of any cellular components like blood cells, but otherwise it has an identical composition to the plasma. This graph shows that the rate of filtration of the molecules across the basement membrane, but if they have the same charge, it is inversely related to their molecular weight. So the molecules having a molecular weight of less than 7,000 daltons will have maximum rate of filtration, just like water, as you can see, around 100. But as the molecular weight increases, the rate of filtration will decrease, and at around 70,000 dalton and above it, the rate of filtration becomes completely insignificant. So it means that if the molecules have same charge, their filtration rate is inversely related to their molecular weight. In the next section, we will discuss about the glomerular filtration rate, or GFR.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.